You're welcome, Olutolu. Olutolu, you're welcome. Welcome to the class. Welcome, Amidat.
Thank you, sir.
Since we're a bit late, it's like I will start the class immediately so that others can join us along the way. Olutolu, are you with us?
I'm with you, sir.
I
You're
can't
welcome,
seem
sir.
to hear.
I'm here,
All
sir.
right. Thank you. I hope the network will not be bad because I'm in a remote place. The network where I am now is not really encouraging. I hope it will not disappoint us.
Amen.
Welcome, Simply Precious.
Thank you, sir.
Most welcome. Let me share this slide. Let me share. Can you see my slide, please?
Yes, sir. Yes.
You can see your slide.
So, Okay, if you could recall, in our last class, we concluded the TCA cycle, which we learned about the last 5 steps of glycolysis, where we saw how the two molecules of glyceraldehyde 3-phosphate is sequentially converted to 2 molecules of pyruvate. And we learned that during that process, 4 molecules of ATP are in turn produced. And the net ATP generated throughout the glycolytic pathway is 2 ATP, because initially at the priming stage, or we may call it at the energy utilization phase, 2 molecules of ATP were initially consumed. The first one was first consumed when the glucose molecule that starts the reaction, that is the substrate of the glycolytic pathway, was energized by reacting with ATP. And that reaction, we said, is phosphorylation, is addition of phosphate group from ATP to ordinary glucose molecule, 1 glucose molecule. And the addition of phosphate group from ATP simply means utilization of energy, and the phosphate group attached to the glucose molecules makes it to have energy to continue the second reaction. Then in the third reaction, another ATP was also added, so that we discover that 2 phosphate groups were added at the end of third reaction of glycolysis, so that you have glucose 1,6-bisphosphate. And we say these 3 steps of reaction are called energy utilization stage, or priming stage, or energy priming stage, while the last 5 reactions comprises of energy generation stage. One of you was asking, one of you asked me, or let me say, few of you asked me how we can summarize this glycolytic pathway, which I said I brought out the chemical reactions involved. That if you're able to know the chemical reactions and just place them the way they are, you have been able to summarize the glycolytic pathway. Yes, you can as well summarize the glycolytic pathway with this slide. This last slide I have here, slide number 24. This is the summary of glycolytic pathway, of which we say glycolytic pathway comprises of 10 reactions, 10 reaction steps. This is summary, but then this summary does not involve the definition of glycolytic pathway. So you may start by defining what is glycolysis from this, and say it involves 10 reaction steps. Here we'll explain that the first 5 steps comprising the preparatory phase, or energy utilization steps, which takes 1 molecule of glucose. That is, it starts with 1 molecule of glucose and produces 2 molecules of glyceraldehyde 3-phosphate. Let me put this GADP in brackets so that we know that it is the abbreviation. It's the abbreviation of glyceraldehyde 3-phosphate. You can write it like this. You can write GA3P. So different texts abbreviates it in different ways. Now let me move this one down here. So this is so this we learned that this first 5 steps utilizes 2 ATP molecules. That is, the cost of this first 5 reactions of glycolysis is the use of 2 energy currency. 2 energy currencies, that is 2 ATPs. Then the other 5 steps makes up the payoff phase, or the energy generation step and stage. Or let me say steps. You can't say stage, say steps. Bracket. In which each molecule of glyceraldehyde 3-phosphate is further converted to pyruvate and lead to production of 2 ATP each in the process for a total of 4 ATP. That is, the net energy production from one molecule of glucose is 2 ATP after minusing or subtracting the first 2 ATP used. Then say the reactions involved also convert one molecule of glucose, which is a 6-carbon compound or a 6-carbon sugar, because there are other sugars that are not up to 6 carbon. Some are 3, some are 5. Then conversion of 6-carbon sugar to 2 molecules of pyruvate. And pyruvate, you know, let me put it in brackets so I know. 2 molecules of pyruvate. Okay, let me see. A 3-carbon compound. Pyruvate is 3-carbon compound. So you can say ATP is the conversion of 1 molecule of glucose to 2 molecules of pyruvate. That is a 6-carbon sugar or a 6-carbon compound to 3. Hear me, please. Can you hear me, please?
Yes, sir, we can hear you.
All right,
I
thank
can
you
hear
very much. Thank you. Nice chat. Then we say the main thing to remember in glycolysis is that glucose in the cytoplasm. Remember, glycolysis takes place in the cytoplasm of the cell. And if you could recall, right from the beginning of our class, we said we are learning those biochemical reactions that happen inside the cell. For what reason? For the purpose of maintaining life to continue as life. For the purpose of not allowing life to go to a standstill. The reason why these biochemical reactions are taking place inside the cell. And as soon as these biochemical reactions continue taking place inside the cell will continue living. And as soon as the cell is alive, then the whole organism is alive. So don't forget these facts. So the main thing you should recall in glycolysis is that glucose in the cytoplasm of the cell is converted to pyruvate. And then as soon as the glucose is converted to pyruvate, the reaction doesn't stop there. And this reaction is only called an anaerobic reaction. This reaction is an anaerobic reaction. I'll ask you, what do you understand by anaerobic reaction? Can someone tell us? Can you come again, please? Lovett?
Yes, sir. I said
Lovett?
anaerobic is. Yes, sir.
Yes, yeah, continue.
I said, is it the reaction that takes place in the absence of oxygen?
Oxygen, yes, you are correct. You are correct. The glycolytic pathway is one of those biochemical reactions that starts way back at the beginning of life, which means that this anaerobic reaction can occur in any kind of cell, whether it is a prokaryotic cell or a eukaryotic cell. Yes, glycolysis is a biochemical reaction that can happen in any kind of cell, whether aerobic or anaerobic, and which form the basis why the microorganisms can survive in any environment where there is air, where there is glucose, or where there's any compound that can be converted to glucose, whether there is presence of air or not, that is, whether there is presence of oxygen or not. So glycolysis is an ancient biochemical reaction that happens in any cell. It's a reaction that is as old as our ancestors. It's an ancestral reaction. In fact, some scientists explain or argue that glycolysis is is a biochemical reaction that follows the evolution of life, that comes or accompanies the evolution of life. So during the evolution of life, glycolysis was there to maintain the integrity or the life processes of cell in as much as energy utilization is concerned. So glycolysis can happen in any kind of living thing. Any living things, provided it's alive, whether there is oxygen in the environment or not, glycolysis will happen in order to sustain that life. Is that understood? Any question on this? So don't be surprised that when we say there are microorganisms inside the gastrointestinal tract, inside the GI tract of living things, of man, for example, in our GI tract or our digestive system, there is a part of our digestive system called colon. Over there, real digestion of food don't take place because the enzymes responsible for digestion, only few of them can survive there, and the condition over there is not favorable to any other microorganisms that does not have the characteristics of that environment. And that is part of our GI tract where non-digestible carbohydrates are stored. And in the process of storing non-digestible carbohydrates over there, things happen. And one of the first things that happen is that the non-digestible carbohydrate that enters that place serve as food to the microorganisms there, because the microorganisms inside that place, they contain or they secrete enzymes that can gradually break down those non-digestible carbohydrates known as fiber, dietary fiber. And when they break them down, they form other compounds different from carbohydrates. That I may not necessarily go into it in detail, but I'll just mention it at this junction, except if any of you have a clarification later at the end of the class, you may ask, because it's beyond your class. So glycolytic pathway is an ancestral biochemical reaction that happens under anaerobic condition, that is in the absence of oxygen. Okay, which is says which then moves what was I about to explain? The glucose in cytoplasm of the cell is converted into pyruvate, which will then move on to the next stage of cellular respiration, which is TCA cycle. So the TCA cycle I'll just summarize what I've just finished explaining now. TCA cycle occurs in anaerobic condition. That is in the absence of oxygen. And can occur both in prokaryotic, let's start with eukaryotic, and karyotic cells. This is what I wanted to add, because it's very important for you to know. So by the time you are able to digest this slide alone, you have been able to summarize whatever that happened in TCA, I mean India, yeah, in the glycolytic pathway. And like I explained before, this is not the end of glucose metabolism or catabolism. This is not the end of glucose catabolism in the cell, because some reactions will continue in the mitochondria, and the reaction that continues in the mitochondria is TCA cycles, no longer glycolysis, because the enzymes that exist, the enzymes that catalyze glycolytic pathway are present in the cytoplasm. Why the enzyme that can continue oxidation of pyruvates are not in the cytoplasm. So pyruvate will be quickly diffused into mitochondria. Inside the mitochondrial matrix, that is between the membrane of mitochondria, either inside or outside or in between the membrane, that is inside the mitochondria, the pyruvate is further oxidized so that more energy is released, because the only 2 net ATP produced at the end of glycolysis is not enough. If the reaction stops there, it means that the cell is not economical in energy metabolism, because about 10 times, I'm saying 10 times, about 15 times, or let me say 17 times of the energy produced by glycolysis it will be produced at the end of TCA cycle and electron transport chain. So the complete breakdown, like I said before, the complete breakdown of glucose, that is this carbon compound, to release energy and carbon dioxide occurs in 3 pathways. The first one is glycolytic pathway, the second one is TCA cycle, and the third one is electron transport chain, of which I said just like we have learned, we'll see the details of the TCA cycle and some aspect of, details of glycolytic pathway, like we have seen already, then some aspect of the TCA cycle for you to really understand what's happening. But for the electron transport chain, is beyond your level now. Or only mention some statements that qualifies that. So here, let me show you another. Let me share. Let me stop sharing this. But I want to share this. There is this document I want to share. Yeah, this. Can you see my slide, please? I mean, can you see my screen? Because it's not slide.
Yes,
Can you see my
sir.
screen now? Okay, good. In another way, I put that TCA, that glycolytic pathway, I put it in a different English here. You see, the English here may look friendly and more familiar to you than the one in my slide. When I'm sharing the material, I'll share this alongside with you, because here the glycolysis is actually summarized, and you see the steps here are more explained, because you see over there in the slide, the caption doesn't show what happens in the reaction, but here you see the first reaction is phosphorylation. But over there in the slide, I say it's utilization of ATP. And utilization of ATP simply means phosphorylation, use of ATP to supply one phosphate group to glucose. So I term that as the first step here, and it's the same thing. In any way, it's the same thing because it's the same reaction. So I name it step. In step 2 is isomerization reaction. And that's exactly what happened. Whatever explanation is going to be given is this isomerization. In step 3 is second phosphorylation. But yeah, let's say first phosphorylation here. Anyway, no need of saying first, because it's the first one, it's the very first reaction. In the reaction number 3, where we use the second ATP, it is also the second phosphorylation. Then reaction 4 is the breakdown of glucose 1,6-bisphosphate. That is the molecule, the end product of second phosphorylation. When you break it down, you now have 3 carbon compound with 1 phosphate group each. That is what happened in reaction number 4. Then in reaction number 5, the second 3 carbon compound is converted to the first 3 carbon compound, which is conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, which is isomerization, because they are all isomers. I mentioned it in my previous class anyway. You can see all the reactions in this note. This is what I'm showing you now. I named the steps by the name of the reaction that happens there. Step number 6 is oxidation. Step number 7 is dephosphorylation. That is removal of phosphate group now from the 3 carbon compound to form ATP. Reaction number 8 is phosphate transfer in the same 3 carbon compound from position 3 to position 1 and to position 2, as you can see here. Then reaction number 9 is dehydration, removal of water molecule, while the last reaction is second dephosphorylation. That is the formation of ATP by utilizing the phosphate group attached to the 3 carbon compound. That is the end of glycolytic pathway. And you can see the cycle here. All what we have been saying, this is it. In a simple structure, you have it. So that if you are able to have this, I will give you a plus. Anybody that can be able to give this reaction step by step, it means the person is not just a pathfinder. The person must have really went through a kind of study before getting into this program. And this table you are seeing is also further making is to further make the reactions simpler for you. You see, I divided it into 4, and these are the 10 steps. I divide each step to 4. I put the reactant, which is the starting material. I put the product, which is the end product of each reaction. For reaction number 1, the starting material is glucose. The end product is glucose 6-phosphate, and the enzyme that catalyzes the reaction is hexokinase. But I told you can also use glucokinase if it's glucose as a starting material. Then further details is here. There's use of ATP. So energy is used up in this reaction, and this has shown that 1 ATP is used up. Second reaction is different from first reaction because the end product of first reaction is used in second reaction as a starting material, and the end product of second reaction is different from the end product of first reaction. And you can see the enzyme too. You can see all of them. The enzymes are different. So this is also a summary of the glycolytic pathway. Anybody with any questions so far on this glycolytic pathway? Because now I want to jump into TCA cycle because it's the next reaction involved. TCA cycle. Anybody with question on glycolytic pathway? Yes, I'm listening, please. Yeah. Love it. Love it, I cannot hear you. Love it, are you speaking?
Yes, sir.
All right, can hear you faintly now.
Can you hear me?
Now I can hear you.
Yes, sir. I said what can be the effect of any malfunction of any enzyme in this glycolytic pathway to our health?
Yes, any of the reaction step, if it's missing, if any problem happen, maybe as a result of anything can happen anyway, and which may be due to many reasons. Whatever case, anything that happens in any of these reaction steps can lead to a disease state. So we are going to discuss disease conditions as a result of any problem of any of these reaction steps. We're going to discuss that after I finish TCA cycle, and maybe after I've discussed lipid metabolism with you, so that We'll discuss all the diseases together because some of the diseases are similar. So if there's any problem with any of the reaction steps, there will be a problem to our health. The first thing that will happen is that it's either the glucose is not utilized, that is at the starting reaction. If the enzyme is not there, the glucose cannot be utilized, cannot be converted to the product required in the first reaction. And if that happens, glucose will be concentrated in our body, will be plenty. I mean, it will gather in the cytoplasm, and because it is not used up, it's like you having a lot of money in your bank. You didn't use the money to do anything. The money will keep on piling up. So the glucose will pile up, and as a result, you know what will happen now. Hyperglycemia, then diabetes. So we'll discuss all that anyway. But by and large, if any of the reaction step doesn't happen, the starting material or the end product of that reaction may accumulate and cause problems. So in a nutshell, but it's beyond that, and we'll see that later. I hope you are clear. Love it.
Yeah, thank you, sir.
Most welcome. Any other question? All right. In the absence of further questions, I think it's good we delve into the TCA cycle. The TCA cycle is also known as the Krebs cycle or the tricarboxylic acid cycle. TCA means tricarboxylic acid, then cycle. So TCA cycle is a continuation of a glycolytic pathway. TCA cycle is a continuation of glycolytic pathway because TCA cycle is part of catabolism of glucose. It's part of catabolism of glucose. Glucose is not completely catabolized or broken down if it does not if it does not pass through TCA cycle, because the end product of glycolytic pathway, which is pyruvate, is not the end of it's not the end of glucose catabolism. So you should have it at the back of your mind. I may ask you during your exam, is pyruvate the end product of glucose catabolism? Or just keep quiet. If any of you was in the attendance in this class today, you quickly recall by saying no, and possibly you give your reason. The reason why it's not the end product is that this reaction of the reaction of glycolytic pathway occurs in anaerobic condition without oxygen, and so therefore only 2 ATP are generated. And if the reaction, that is one. Secondly, if the reaction stops at pyruvate only, it means that the use of energy of glucose is not economized. Glucose is not maximally breaking down because glucose can produce about 15 to 17 times the energy that the glycolytic pathway can produce. Because at the end of glucose catabolism, 36 to 37 ATP will be generated, whereby about 34 to 35 ATP, net ATP will be generated. You can see, if 35 ATP can be generated, why stop at only producing 2 ATP? It means that it is no economy. The body cannot economize glucose utilization. And finally, or let me say the third reason why TCA cycle, why pyruvate is not the end product of glycolytic pathway is that when TCA cycles when pyruvate enters into TCA cycle, pyruvate is not stable. There's another reason why pyruvate is not the end product of glycolytic pathway is that pyruvate is not stable in the body. It can easily be converted to other products. Then fourthly, pyruvate, when it enters into TCA cycle, it can easily produce other compounds called intermediate compounds or other metabolites that can be be useful to other pathways that can be used as starting material for other biochemical reactions. Let me come again. It is not there in your slide. It is because of my explanation that this came to my mind. So I'm mentioning it now. So take note. I said, if you are asked, why is pyruvate not the end product of glucose catabolism? One, pyruvate is not the end product of glucose catabolism because, one, the reaction occurs in anaerobic respiration. That is TCAs and carboxylic acid cycle. Sorry, glycolytic pathway occurs under anaerobic condition. That is one. Meaning it can have a chance to express its aerobic reaction, which is further use of pyruvate. Two, pyruvate, if it's an end product of glucose catabolism, it means that the glycolytic pathway or the body, let me say, the body cannot economize energy metabolism from use of glucose because it's going to waste about 30-something ATP that is supposed to generate when it goes further beyond formation of pyruvate. Thirdly, pyruvate is not stable, so it can easily be oxidized or converted to other compounds. Fourthly, if pyruvate further move into TCA cycle, it can be used to generate other intermediate compounds that can be used as starting material for other biochemical reactions, meaning it is a raw material for other compounds. So these four reasons only, or let me say among others, can serve as the reason why pyruvate is not the end product of glycolytic pathway, and it will never be the end product of glycolytic pathway. Professor is asking me to add it to the notes. You see, it's our lecture time. Sometimes I deliberately don't want to add it to the lecture notes because so that you in attendance will have a plus. But you can see sometimes, even without you asking, I do add it to the notes, right? I do add some of this explanation to the notes without you asking. But at times, I need to keep mute as regards to the notes. But when I say it here and it talks to your brain, or when you go further to listen to the recording, you understand it better. Yes, that will be a plus to you that listen or attend the class. So the more reason why, yeah.
Sir, please, what brings about pyruvate?
What brings about pyruvate?
Yes, sir.
Pyruvate is the end product of glycolytic pathway, and we saw it in our last class.
Yeah, but you said now that it can never be the end product of glycolytic pathway.
What I mean is pyruvate cannot be the end of the reaction of glucose catabolism. Did you hear what I said? I said glucose catabolism, not the end product of glycolytic pathway. At the end of glycolytic pathway, we form pyruvate, but the catabolism of glucose can never stop at pyruvate. And I gave these four reasons why it can never stop there. Is that clear, Nurad?
Yes, sir. It's a bit clear, sir.
No, it's not clear. I can see it's not clear.
Yes, sir.
Okay. Did you listen to the part when I said the glycolytic pathway is one of those biochemical reactions that accompanies the evolution of cell? Did you listen to that?
No, sir.
That means you came in late. We say glycolytic pathway occurs in anaerobic condition. So for that reason, it does not only happen in eukaryotic cell, but it also happens in prokaryotic cell. Remember, we are dealing with eukaryotic cell. That is the most advanced cell now. We are dealing with the cells in the human body. But at this point, we said, or we learned that the glycolytic pathway can also occur in prokaryotic cell. I think you'll be surprised that, but we said eukaryotic cell is the most advanced cell. How can the reaction in most advanced cell happen in the primitive cell like prokaryotic cell? So I gave the answer myself, even without you asking. I said the glycolytic pathway is one of those biochemical reactions that accompanies the evolution of cell. Even before the existence of cell, this reaction occur because it's the reaction that makes energy utilization possible in primitive cells before they evolve during evolution into the most advanced cell. So it can happen in the absence of oxygen. The reason why it's called an anaerobic reaction. All the reactions in glycolytic pathway are anaerobic. They are in the absence of oxygen. They occur in the absence of oxygen, so they are anaerobic. So for this reason, they are reactions that whether there is oxygen or not, it happens and it can allow the cell to survive wherever the cell is. That is one. Then secondly, we say the glycolytic pathway is the breakdown of a 6-carbon sugar, that is glucose, into a 3-carbon sugar, that is pyruvate. And the reaction involves 10 steps of chemical reactions. I hope Nurat is listening to me. We say the glycolytic pathway is a 10 reaction steps
Yes, sir.
that involve, yes, it is a 10 reaction steps that involve the breakdown of glucose. Remember, we said, or we have mentioned that glucose catabolism ends in formation of carbon dioxide and ATP. And for that to happen, it must follow through the glycolytic pathway, the TCA cycle, and the last one, which is the electron transport chain. But I will give more emphasis on glycolytic pathway and TCA cycle because that will make complete utilization of glucose. If it does not follow these 3 steps or these 3 pathways, complete utilization of glucose is not achieved. So the reason why we say formation of pyruvate is not the end product of, or it's not the end of glucose catabolism. Remember, I said glucose catabolism. I did not say it's not the end product of glycolytic pathway. No. I only said it's not the end of glucose catabolism in energy generation. For energy to be released from glucose, pyruvate is only a starting material because it's going to be used to start the next cycle. That is the glycolytic pathway. So pyruvate is only the end product of glycolytic pathway, but not the end product of utilization of glucose to produce energy, or let me say, not the end product of glucose catabolism. I hope that is understood. So when pyruvate is formed, 2 net ATP are generated and released. And when that happens, pyruvate will now be further converted to acetyl coenzyme A, called acetyl-CoA, as you are seeing here, as you've seen in this material I'm sharing. Pyruvate is quickly converted to acetyl-CoA so that TCA cycle can easily be initiated. So that it can be easily it can easily initiate the TCA cycle. So when pyruvate is formed, if it does not quickly converted to acetyl-CoA, then TCA cycle is not initiated, cannot be initiated. So it must be converted to acetyl coenzyme A because acetyl coenzyme A or acetyl CoA is the starting material for TCA cycle. So the Krebs cycle takes place in the mitochondria. As soon as pyruvate is formed, it is channeled into the mitochondria of the cell and converted to acetyl-CoA in the mitochondria of the eukaryotic cell. It's in the mitochondria of eukaryotic cell, that's where this reaction happens. But in prokaryotic cell, it happens in the cytoplasm, which means that Krebs cycle or TCA cycle can also occur both in prokaryotic and eukaryotic cells. So don't say I told you that TCA cycle does not occur in prokaryotic cell. Just like the glycolytic cycle, the TCA cycle also occur in the prokaryotic cells. And for TCA cycle to start, the pyruvate has to be produced. Production of pyruvate from glucose is called preparatory step for TCA cycle, which means the glycolytic cycle prepares the glucose to form pyruvate, or prepares the starting material for TCA cycle. So glycolytic pathway is only meant to prepare the starting material for TCA cycle. So pyruvate is derived through glycolysis. That is from breakdown of glucose to pyruvate. Now, for TCA cycle to start, the pyruvate needs to be oxidized to acetyl-CoA by the enzyme called pyruvate dehydrogenase complex. That's the enzyme. And in this reaction, a molecule of carbon dioxide and a molecule of NADH is generated. The acetyl-CoA is a 2-carbon compound. In the next step, the acetyl-CoA combines with oxygen as it combines with oxygen, and this is a nutshell, so to say. It combines with oxygen to produce some intermediates. It combines with oxygen in the presence of an enzyme, which is a different enzyme entirely, to produce a 4-carbon compound known as citrate. And citrate is the first product of TCA cycle. It's the first end product of TCA cycle. That is first end product for the first reaction of TCA cycle. And I will not want to expand this TCA cycle beyond this. So even when studying, you are only going to read TCA cycle and stop in these first two paragraphs. Because these first two paragraphs are, to some extent, talks much about what we need to know. But let me see the end. Does it? Okay.
Sorry, sir.
Yeah.
Sorry, what did you say is the end product of TCA cycle?
No, I didn't say end product of TCA cycle. I only
No, you
mentioned.
just said the first end product for TCA cycle is.
No, the first end product for the first reaction of TCA cycle.
Okay.
The end product for the first reaction of TCA cycle. Because TCA cycle also involves some steps of reaction. And these steps of reaction are 8. There are 8 steps of reaction. They're not up to 10 that we have seen. We have seen 10 reaction steps in glycolysis, right? But in TCA cycle, there are only 8 steps of reaction. So the first step produces
the first end product of the first reaction of TCA cycle. What is this?
That is citrate, C-I-T-R-A-T-E. Citrate, C-I-T-R-A-T-E, citrate.
Okay, sir. Thank you.
You're welcome. So the results of Krebs cycle, that is TCA cycle, The products of TCA cycle are 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of GTP. Remember last time I said GTP is an isomer of ATP. GTP means guanosine triphosphate. Just like how we have adenosine triphosphate as ATP, we also have GTP. So, and GTP is also an energy-rich compound, just like ATP, but it cannot be utilized by the body until it's converted to ATP because GTP and ATP are isomers. So, the end products of TCA cycle as a whole is what I'm saying now, is this last paragraph you are seeing. That is 3 molecules of NADH, that is nicotinamide adenine dinucleotide in reduced form, that is together with hydrogen, NADH. 1 molecule of FADH2, 1 molecule of GTP, and 2 molecules of carbon dioxide. 2 molecules of carbon dioxide. Now, since glucose is split into 2 pyruvate compounds, for each molecule of glucose, this cycle runs twice. Hence, the products formed are times 2 of each. For example, the 3 molecule of NADH times 2, making 6 NADH. 1 molecule of FADH2 times 2, making 2 molecules of it. 1 molecule of GTP times 2 means 2 molecules of it. And 2 molecules of carbon dioxide means times 2. That is 4 molecules of carbon dioxide. So this paragraph summarizes the TCA cycle. You should understand this paragraph very well. Hello, can you hear me now? My network is shaking.
Yes, sir.
All right, thank you. My network was shaking earlier and ticking off. Though this is let me scroll down. Okay, let me share. This is summary of the, let me put this in bold. This one will teach you, will make you to recall that these are the end products of the TCA cycle. The TCA cycle did not produce only one end product like the glycolytic pathway. I think you can see the difference between TCA cycle and glycolytic pathway, right? The glycolytic pathway only have one end product, which is pyruvate, but 2 molecules of pyruvate. But the TCA cycle, why the glycolytic pathway produce only pyruvate, that's 2 molecules of pyruvate, the TCA cycle have about 1, 2, 3, and 4 end products. It has 4 end products. And among those 4 end products, one of them is 6 molecules of it. That's 6 molecules of NADH, 2 molecules of FADH2, 2 molecules of GTP, and 4 molecules of carbon dioxide. And what you should also know is that, look at the last statement after this last paragraph. You say all the NADH and FADH2 are fed to the electron transport chain for the generation of ATP, meaning the 6 molecule of NADH and the 2 molecule of FADH2 are not just produced, they are also further used to produce ATP. They are further used to generate ATP. Let's assume we have 2 ATP generated at end of this TCA cycle. Then the 6 molecules of NADH and 2 molecules of FADH are further, that is their next reaction is they will enter another step of biochemical reaction, which I said is electron transport chain, in order to maximize energy utilization from glucose so that they produce glucose. Because this NADH and FADH2 are called reducing equivalents. They are called reducing equivalents. Let me put it here. E times New Roman 12. So the NADH and FADH2 are known as reducing equivalents. And are rich in energy, which can only be harnessed through the electron transport chain, ETC. That is the meaning of electron transport chain. So because the NADH and FADH2, all of them have energy they are carrying. They carry energy, both of them. All of them carry energy because NADH produces about, each molecule of NADH produces about 9 ATP or so, while each molecule of FADH2 produces about, is it 5 or 6 ATP? Then if you multiply it by that, by the number we have here, you see we'll have about 30 something ATP. Because at the end of glucose catabolism, that is the amount of energy generated. So these last 2 paragraphs, they really explain the end product of the TCA cycle. So let me show you this slide that explains, that tries to explain the TCA cycle. There is this slide. So like today I'm giving you too many stuff, but I just need to do that so that we can cover. Also, I can cover some things today, even though we started late. So share. I'm trying to share this cycle. Can you see my screen now?
Yes, sir, we can see it.
Thank you, Precious. So here you can see the TCA cycle. This is the summary of it. The TCA cycle It's called tricarboxylic acid cycle. Call it Krebs cycle. It's also it is one of the central metabolic pathways that connects different biochemical pathways, like I have mentioned before. I say it's an example of amphibolic pathway because you can neither classify it as catabolic pathway or anabolic pathway, or else just put it, you can put it inside anabolic pathway and you can put it inside catabolic pathway. You are correct if you do any of them. So TCA cycle is one of the cycles that involve glucose metabolism, whereby glucose is oxidized to form pyruvate, and which the pyruvate is now channeled into the TCA cycle when oxidized to acetyl-CoA. It is the gateway to aerobic metabolism. So TCA cycle is that biochemical set of biochemical reactions that channel the product of anaerobic reaction to aerobic reaction. So TCA cycle is an aerobic reaction, not anaerobic. It occurs in the presence of oxygen, unlike the glycolytic pathway, which occur in the absence of oxygen. The tricarboxylic acid cycle is a series of chemical reactions used by all aerobic organisms to release stored energy through the oxidation of acetyl-CoA derived from carbohydrates or fats and proteins into ATP and carbon dioxide. You can see what this definition is telling us is that the TCA cycle can utilize both carbohydrates, fats, and proteins. The reason why TCA cycle, one of the reasons why TCA cycle is called amphibolic pathway. It does not use only one nutrient. It can use carbohydrates, it can use fats, and can use proteins. And all of them can get into it. And from this TCA cycle, you can get metabolites or intermediate compounds that can be used to synthesize carbohydrates, that can be used to synthesize proteins, and that can be used to synthesize fats. So you can see you can find TCA cycle to be both anabolic and catabolic. So in between them, you can just give a name. I mean, a name is given to be called amphibolic. The reason why you cannot say TCA cycle is a catabolic pathway only or anabolic pathway only, but you can say it's an amphibolic pathway. This diagram is showing you. You can see carbohydrate can be converted to acetyl-CoA. Protein also can be converted to acetyl-CoA. Fats can be converted to acetyl-CoA. And as soon as acetyl-CoA is formed, the best reaction it can undergo is to enter into TCA cycle. But acetyl-CoA can be converted to other compounds too. But left alone for the human body, it loves converting acetyl-CoA into TCA cycle to produce energy, which is a natural process. And it's a process that is required by every cell. Every cell of the body requires this reaction because it generates energy for the body to utilize. So the Krebs cycle takes place in the cytosol of prokaryotic cell, but it takes place in the mitochondria of eukaryotic cell. So in the human body, these reaction steps occur in the mitochondria, not in the prokaryotic cell, not in the cytosol, sorry. The net result is the production of carbon dioxide when acetyl group from acetyl-CoA enters the TCA cycle. So, why is TCA cycle called amphibolic pathway? It is quite obvious that there are both catabolic and anabolic pathways in TCA cycle. So it is not easy for you to say TCA cycle is an anabolic pathway or a catabolic pathway. So you'll be wrong to say TCA cycle is only a catabolic pathway or only an anabolic pathway. No, This is not so. And this is the reason why it is called amphibolic pathway, or one of the reasons why it is called amphibolic pathway. Because in it, there is synthesis of very important compounds that can be channeled to production of other compounds. For example, succinyl-CoA is one of the intermediates of TCA cycle, and it is required for synthesis of red blood cells and synthesis of red blood itself. So the heme protein present in the blood is synthesized from succinyl-CoA. Oxaloacetate is also a molecule as an intermediate in the TCA cycle that can be converted to glucose through gluconeogenesis. And it also can be converted to amino acids for the synthesis of protein. Nurat, are you asking a question? So I want us to stop here. Do you have any question?
Yes, I have a question.
Love it. Carry on.
So I have a question.
Carry on.
Yes, sir, please, I want to be cleared on that citrate. You mentioned the citrate.
Citrate is the first end product in the reaction that starts the TCA cycle. Yes.
So, sir, I don't know if I'm correct. It's
citrate
not,
is the first product produced in TCA cycle. If you say so, you are correct. It is the first product produced in TCA cycle.
So I can say that oxaloacetate combines with acetyl-CoA to form citrate. Am I correct?
Yes, you are correct. Okay. Very, very correct. Thank you for that question. I know some of you must have not seen anything oxaloacetate here. Oxaloacetate is already existing inside the cell. As soon as acetyl-CoA is formed, like I told you, it does not stop. So the signal that acetyl-CoA produces is that TCA cycle is ready to occur, because before the production of TCA of acetyl-CoA, TCA cycle wasn't in existence, was not happening already, because oxaloacetate is already there since waiting, but because there is no the other starting material. Oxaloacetate and acetyl-CoA are the starting material, two starting material that react to start the TCA cycle. But oxaloacetate has been waiting for a long time without acetyl-CoA. So when the body is ready to undergo TCA cycle, acetyl-CoA is formed. As soon as it's formed, the second reactant, which is the second starting material, that is the oxaloacetate, will quickly react with acetyl-CoA, and citrate is formed as the first product of TCA cycle. And let me tell you, the first product of TCA cycle is acetyl-CoA, while the last product of TCA cycle is oxaloacetate. That is another thing you should know. The first product of TCA cycle is citrate, and the last product of TCA cycle is oxaloacetate. And that last product is also one of the starting material for the TCA cycle. You can see how nature is being economical, very, very economical, where the end product of a particular reaction pathway is one of the starting material for that same pathway. So any other question? Yeah, I'm waiting for questions, please. So I guess no any other question. In our next class, we'll start with the importance of Krebs cycle. I'm going to check why Krebs cycle, why TCA cycle. I'm going to see the importance. So at this point, I will stop recording I will share this slide with you and the previous slide, and possibly with that material that I have also shown today, so that you can go through and you can be able to ask serious questions in our next class. Thank you for listening and appreciate your time.
Thank you very much, sir.
Thank you very much, sir. We sincerely appreciate you, sir.
You're welcome. Thank you for listening.
Thank you.